let us discuss about the seed. Seed is the result of sexual reproduction. Here the seeds are developing after fertilization. After fertilization, the ovules develop into seeds. The seed is having mainly two parts here, seed coat and embryo. Here it is the seed, and the seed is having the seed coat. Sometimes it is hard, sometimes it is very thin, and having two layers, outer layer and inner layer. Outer layer is called as testa and inner layer is called as tegmen. It is outer layer, and tegmen is inner layer. And inside the seed coat, here embryo is present. Here embryo, and here embryo is having two parts, mainly embryonal axis. Embryo is present here. Embryonal axis and cotyledons, two parts are there. Here embryo and two cotyledons, one or two cotyledons. One or two cotyledons. One or two. So one cotyledon is present in monocot plants. In monocot plants like here wheat, maize, rice. So in these crops are in Poaceae family members are in monocot plants. Here the cotyledons only one cotyledon is present. And in dicot plants like mustard or pea. So in these plants two cotyledons are present. Here one cotyledon or two cotyledons. So it is dicot. For example, it is dicot seed. Dicot seed is having two cotyledons. It is one cotyledon. It is one cotyledon. Two cotyledons. And generally the cotyledons are rich in nutrients, rich in proteins, or rich in nutrients. And here embryonal axis. So in the embryonal axis is having here two parts. One is a plumule and the second one is radicle. So in the embryonal axis, the upper part or which develops into shoot system, that is called as plumule, and lower part, that is radicle. Radicle develops into root system. So the two parts, upper part or the embryonal axis ends with a plumule and a radicle. A plumule develops into shoot system, radicle develops into root system. So here in the seed mainly two parts, seed coat and embryo. Seed coat is outer layer, embryo is here having two parts, mainly embryonal axis and two cotyledons. It is embryonal axis and two cotyledons. In the embryonal axis, it is ending with plumule and other end is with radicle. Cotyledons one or two. In monocot plants, only one cotyledon. In dicot plants, two cotyledons. So based on the number of cotyledons only, the angiosperms are classified into here two types: monocotyledonnae, dicotyledonnae. Monocot plants, dicot plants. And here if we see the here dicot seed, first of all here we are going to discuss about dicot seed or structure of the dicot seed. So in the dicot plants, here the embryo is having two cotyledons. So it is the embryonal axis. The embryonal axis, one end is with plumule. Embryonal axis ending with plumule, or this plumule is developing into shoot system. So plumule develops into shoot system. and it is the radicle. The radicle develops into root system. And these are the cotyledons. Cotyledons, and the cotyledons are rich in nutrients or proteins. In pea plant or Fabaceae members, leguminaceae family members, the cotyledons are rich in proteins, or they are storing the proteins. And here plumule, radicle, and two cotyledons completely is embryo structure. And in the here plumule and radicle, these are not covered by any sheaths. Here plumule is developing into shoot system, radicle is developing into the root system. Here two cotyledons are present, and these two cotyledons are rich in nutrients or sometimes proteins are present. Here endosperm, in the generally seed, here endosperm is also present. So endosperm is the result of a double fertilization. In angiosperms, here double fertilization is observed. Double fertilization means two fertilizations. So due to the first fertilization, zygote is formed, zygote develops into embryo. And due to the second fertilization, here the endosperm is developing. Endosperm is here sometimes in the seed endosperm is present, sometimes it is absent. If the endosperm is present, that type of seeds are called as endospermous seeds. And if the endosperm is absent, non-endospermous seeds. Endosperm is present in some seeds like in castor. In castor plant, endosperm is present. So here after the development of the embryo also some endosperm is remaining. So the remaining endosperm is, it is if the endosperm is present in the seed, that is called as endospermous or albuminous seed. And non-endospermous seeds, generally dicot seeds are, they are not storing the endosperm. Non-endospermous seeds, the endosperm is completely utilized after the maturity, after the development of the embryo and seed, endosperm is completely utilized. So if the endosperm is completely utilized, there is no endosperm in the seed. So here these are pea seeds. So these are some examples, non-endospermous seeds. Endospermous, non-endospermous. So it is about structure of the dicot seed. Two cotyledons, embryonal axis, plumule, radicle, and in seeds sometimes endosperm is present, sometimes absent. If the endosperm is completely utilized, no endosperm is present, like in pea plant. And sometimes endosperm is not completely utilized. Some amount of endosperm is remaining. So example, castor. Let us discuss about here structure of the monocot seed. Let us discuss about monocot seed structure. Here in monocot seeds like rice, wheat, maize plants, here only one cotyledon is present. So mainly here seed coat and embryo, only two parts are present. Here seed coat, here if you see the wheat or rice, here seed, in the rice or wheat, the fruit is dry indehiscent fruit. So in this fruit, the pericarp is very thin, and it is fused with the seed coat. Pericarp is fused with the seed coat. So completely it is fused. It is pericarp and seed coat both are fused. Next, inside the major portion, major part is occupied by endosperm. So endosperm is present here. So outer layer is pericarp. Here seed coat. So both are fused. The major portion is endosperm. So in monocot seeds, the endosperm is occupying the major part and it is nutritious or storing food material. So endosperm is present in almost all monocot seeds. So endospermic seeds. These are endospermic seeds or albuminous endospermic seeds. Here endosperm is outer layer. So the endosperm is covered by or outer layer. It is called as it is proteinaceous. The proteinaceous layer is called as aileron layer. Outer layer of the endosperm, it is called as aileron layer. Endosperm is covered by aileron layer. It is also part of endosperm but outer layer. Generally endosperm is triploid. Triploid, it is also a triploid. So due to the double fertilization, the endosperm develops. Endosperm is triploid. Triploid means 3 sets of chromosomes are present or 3 genomes are present. In aileron layer also, here 3 sets of chromosomes are present. So it is endosperm storing food material. And in the groove of endosperm, here embryo is present. Here, embryo. Here it is embryo. Embryo is it is located or situated in the groove of endosperm, and it is only having the embryo is having only one cotyledon. Only one cotyledon. That cotyledon is called as scutellum. The cotyledon is shield shaped and very large. Only one cotyledon is present. This cotyledon is called as scutellum. And in the embryonal axis, here plumule, radicle are present. Plumule, it is the plumule and it is radicle. Here plumule and it is radicle. So plumule is covered by here one sheath-like structure. Here radicle is covered by also one sheath-like structure. The structures are, if we observe clearly here, it is cotyledon called as scutellum, and here plumule, it is a plumule. Plumule is covered by one sheath. Here one sheath is present. That sheath is called as, here this sheath is known as coleoptile. Plumule is protected by a sheath. That sheath is called as coleoptile. Coleoptile is observed in only monocot seeds, not in the dicot seeds. And here radicle, it is radicle, develops into root system and it is covered by one sheath. So this sheath is called as coleorhiza. Coleorhiza, it is a protective sheath. and radicle and coleorhiza both are covered by a root cap. It is the embryo structure. This embryo structure, if we enlarge, so it is the embryo. So total seed structure, it is the seed. The seed is having one layer, pericarp. Pericarp and the seed coat both are fused. So the type of fruit is caryopsis. In the caryopsis fruit, pericarp and seed coat both are fused, and major part is occupied by endosperm. Outer layer of the endosperm is alluron layer. Alluron layer is separating the embryo and endosperm. So it is embryo, it is endosperm. In the embryo, mainly two parts. It is embryonal axis. In embryonal axis, and only one cotyledon is present. So this cotyledon is called as scutellum. Cotyledon is called as one large shield shaped cotyledon. It is called as scutellum, and it is a plumule. The plumule is covered by a sheath. Plumule develops into shoot system. It is covered by a protective sheath, that is coleoptile, and radicle is covered by coleorhiza. Radicle develops into root system protected by coleorhiza, and both are covered by root cap. So here coleoptile and coleorhiza both are observed in only monocot embryos or monocot seeds. So this is the structure of monocot seed. So in dicot seeds, two cotyledons are present. In monocot seeds, only one cotyledon is present. Here these are the some differences between the dicot seed and monocot seed. Mainly monocot seeds are here having endosperm. The endosperm is present. These are endospermic seeds. Perisperm. Perisperm ploid is diploid. So what is perisperm? So during the development of embryo or seed, the total nucellus tissue, the nucellus tissue is also utilized by the embryo, but sometimes here the nucellus tissue is remaining. The remnant of nucellus tissue is called as perisperm. Here you know the structure of here ovule. In the ovule, the total tissue is called as, it is called as nucellus tissue. Nucellus is diploid. So during the development of here embryo, here the total nucellus tissue is utilized by the embryo and embryo sac and the seed. But here sometimes the nucellus tissue is remaining. The remaining nucellus tissue is called as perisperm. So the ploidy of nucellus tissue ploid is diploid. That's why here perisperm is nothing but nucellus tissue only. Remaining nucellus tissue. After the development of embryo, the remaining nucellus tissue is called as perisperm. It is also diploid. So here perispermic seeds. Perisperm containing seeds. Example for perispermic seeds: Piper nigrum or black pepper. It is about perisperm. It is diploid, develops, or it is the remnant of nucellus tissue. Example for perispermic seeds: Piper nigrum and black pepper. Piper nigrum or black pepper.